guess we can start the session. So welcome back to the session 3. Our first speaker is, okay, it's on now? Perfect, okay. Welcome back again to the session 3. Our first speaker for this session is Dr. Vishwanathan Dupatla. He's currently the Vice President Biopharma Innovation and Biomanufacturing at IKP Knowledge Park here in Hyderabad. Vishu.
Don't start the timer. So I will take only 5 minutes, and I thought we can have 5 minutes for discussion. I was at ILS speaking with some of the students, faculty members who wanted to start companies. So they wanted to know if my student is having a project, and if they want to apply for any of these grants, how does it work? So it's the same thing. I'm going to present about I have been talking from morning about biotechnology ignition granted posters with students with examples. I took one example of such grant program where a student came with some interesting results from the lab and came for the grant. This is IKP Knowledge Park in the Genome Valley. So we have around 1,500 students, doctors, engineers who established companies through various grant programs, BIRAC being the flagship program. IKP also worked with Bill and Melinda Gates Foundation, USAID, DFID, state governments. I will not get into all these details, how the technology readiness levels, but I thought I will directly take through some of the points what you have to think. Somebody is thinking if they have to start a company, what are the various parameters? Please don't take photos, you will get these slides. So when you're thinking about any project, so whether there is an unmet need, if you make the technology or a product, whether the buyer is there or not there are very important things you have to think. And also the regulatory, be it a medical device, be it a drug, be it a food product, you have to think about the regulatory path. At the posters, it is good that some of you are already aware of these things. And as I mentioned, business viability or the market potential, and the clinical path, in case if you are thinking especially about medical device and drugs. So these are the 4 things you have to think. I will not go through all these 10 steps. So this is for you. If you just type GATE, it will come. Technology ready levels, BIRAC. The website has both for industrial biotechnology. They have a very detailed manner they have prescribed. These are the various support programs. Thanks to Dr. Pakala morning, he was mentioning. So be it BSc or MSc students, this is only I'm talking with reference to the BIRAC, but there are a few other programs. But in the life science, if somebody is thinking about a company, BIRAC is the place. You have to visit the website every week to see for the updates. Starting from 15 lakhs to 700 lakhs, support system is available. The initial funding is in the form of grant. The later ones are in the form of equity. Anybody interested in these things will be happy to talk about those things separately after this talk. And biotechnology ignition grant, which just got closed yesterday. This time it's a very interesting, thanks to DBT, BIRAC, the partnership with India Health Fund. They have funded additional things besides the ones that BIRAC is funding in the same program. Antimicrobial, digital health, tuberculosis. So these things are there, but every time there are some specific updates. When you visit the BIRAC website, you will see. But anything that touches lives. So the name is Biotechnology Ignition Grant. In the morning, somebody was talking about plants, environment. All these are covered. Clean energy. So don't keep that with you. You just ask the program manager. They will be happy to help you with those details. And in this program, you'll get up to 50 lakh support. And if you're a student, you are most welcome. If you're a company, it should be less than 5 years. As simple as that. So this is where, this is a remote place in Karnataka where the student was doing an MSc project, MTech project, and they had seen that 1 fraction of cucumber extract is attracting a lot of insects. And that is when they called that, hey, everybody is saying that government gives money, will we get? Because, we are just students. So that is where we started. Thanks to BIRAC, there are several outreach programs where we interact with students from institutes, universities, any place actually. I visited more than 100 places in the BIRAC program to talk with the students to help them. The success rate is 10%, but It will give you an idea. It will help you improve your CV. So basically, once they have something interesting and they're working on the project, they need to generate some preliminary data. It's very important. You work on the things what you want to work in the future. So when somebody is asking, what next, if you plan your, trainings, skill sets, you can make use of things. And, during my postdoc, I wanted to be a science communicator. I did a course on, handling the LinkedIn, Twitter, social media communication, and you will be surprised the salaries paid to the social communication are like a 3x of scientist. I'm talking about UK. I'm sure in most places it will be the case. Yeah. So once they have seen that fertile fraction is attracting, it's very important, right? In a MSc project, you have a 3 months time. You see very small things, and you don't have resources, but you can always look into the literature. So that is the technology readiness level 1. So for moving on to the 2, you need to reproduce, show the data. What is that is attracting? So you need to work on all the things. And since time is less, I like to keep this as suspense, but I will tell very big. The compound they have identified is called pheromone, which is more powerful than the pheromone, but because it attracts, it's based on the food sensing, and it attracts both the sexes, and it is so stable that the entire season it can stay in the field. So the student was able to formulate, and again when you are going to a committee, as you said, like when we are a student, they will be evaluating us. You need to go with some proof, which is not very expensive experiments. But if you come to Cdfd incubator or any of the BIZ partners, they will be happy to help you with how to formulate these things. I have done this job for last 6 years. I'm continuously doing. I'll be happy to talk with anybody. So when they took this extract to a cucumber field, the filter paper is full of cucumber pests within 5 minutes. In 2 hours, the filter paper broke. That much strong it was. And when they take to a mango garden in other places, nothing. It is clean white, except 1 or 2. So we asked them to go to a entomologist and get it certified, whether it is specific pests, whether any honeybees are there, and they came with such a clean record I had never seen. I couldn't believe because it is so clean. But students they have formulated, they made the doses. But when you're asking next 50 lakhs, this 50 lakhs you got, you have done some trials. But for asking next 5 crores or 10 crores to establish an industry, you need much more, right? Thanks to again BIRAC and NBM, DBT, the NBM National Biopharma Mission. So World Bank supports technology transfer offices across India. So they have lawyers and people who can help. Again, IKP is one of them. We'll be very happy to help when you are coming early stage, be it for industrial partnership or to help in formulating your IP strategy. So basically, when you come with such kind of data, you need to look what is the prior art. So what are the published data? What are the patents, freedom to operate? So we'll be happy to help and formulate. Thanks to the Prime team, IKP Prime is the technology transfer office. They actually gave this initial crude design to them, and they patented across 7 countries. They went to Brazil, 5 best agri teams from India. And within 4 years, I joined in 2018 and 2021-22, they had a 4 crores revenue. That is the last detailed follow-up I have done by visiting and looking at. I'm sure they're doing quite well. They received another BIRAC funding in a Agri Grand Challenge program to double the farmer's income. So when you're not using pesticides and using a pheromone kind of thing, that immediately qualifies for export. And when there is no pesticides on food, the price goes by 3 times. So it's a very simple. Yeah, these numbers are very important. So because somebody is buying you something from you, they should benefit. So this you have to think about your project, and anybody thinking about a starting company, the first step for you. So I don't have time for grant management discussions, but we'll be happy to come. Other day I was at ILS. I have, I think, around 20-25 scientists from various institutes, DBT institutes. We worked and received the grants. Mainly the faculty works very close with you. You will be the owners of the company, and your faculty will be the advisors, but you can work out based on your institutes. I will stop here, and I have another 2 minutes to any Q&A.
Thank you, Vishu. I think probably they will catch you later. So now I request Dr. Muralidharan Bhashyam to present the moment.
There is.
Yeah, please.
Yes,
very quick question.
Yeah, it's a mat bole. Okay, Shubha Narendra from IIT Hyderabad, and two of his students, again, I will tell all BSc examples. They have developed a very nice lateral flow sensing mechanism, and the students have applied, and they received a follow-on 2 crore funding, private funding, and the faculty wanted to take a back seat. Students have the 95%, the faculty as advisors have the 5%. And University of Hyderabad, I was telling about the bikers. There was one professor who is part of this program. Again, with this 50 lakhs, they were able to show it is a very complex biosimilar, which has 44 lysines, and it's a tetramer, and it should be pegylated at only 10 sites, not more than 11, maximum 11, and specific positions are there. They were able to do that, and they received a around 40 crore funding. One faculty and 3 postdocs together. I will be happy to invite them, and I was speaking with your student, and I'm thinking actually taking them, because those are the guys, postdocs, who have done leaving all the things, and there's a full-time thing.
Okay, thank you, Vishu.
Thank
Now
you
I
very much.
request Dr. Mulidharan Bhashyam to present the momentum to Vishu. 1 moment, please. Thank you. We'll quickly move on to the flash talk. Now I request poster from 41 to 45, please come forward.
Good afternoon, everyone. My name is Pragya Keshavani. I am a PhD candidate at RCB Faridabad, working under the supervision of Dr. D. Sundar from IIT Delhi and Dr. Deepak T. Nair from RCB Faridabad. My research focuses on improving the efficiency of CRISPR-Cas system. So while researchers have been using Cas9 for genome editing, but after the discovery of Cas12a, researchers have moved their interest towards Cas12a due to their low off-target activity. But there is a lack of computational resources and studies for Cas12a. To overcome this gap, this study focuses on Cas12a. Despite the potential of CRISPR-Cas system, it has a few challenges such as off-target activity and incorporation of undesirable repair outcomes. So to overcome the off-target activity, we developed a machine learning-based pipelines to design an optimal guide RNA by predicting off-target sites and their efficiency, because not all the off-target sites present in the human genome are going to cleave under experimental conditions. Also, we studied the role of biological important features in the classification of targets. Along with that, we wanted to understand the recognition and the target binding mechanism of Cas12a, which can also affect the off-target activity. So we studied the change in conformational dynamics of different variants of Cas12a and their bound guide RNA DNA hybrid, and along with that, we have also focused on understanding the role of local sequence context in the in incorporation of undesirable repair outcomes. For that, we developed a pipeline to predict the repair outcomes by employing deep learning based model, which uses attention mechanism and that learns from the target sequence and their flanking regions to predict the repair outcomes. Thank you. My poster is placed at 41 position. You are always welcome to visit my poster. I will be discussing in details there. Thank you.
Good afternoon, one and all. This is Pratigya Dash from Institute of Life Sciences, Odisha. And today I'm going to give a glimpse of the basic ideation of my thesis entitled Crafting the Surfaceome of Osteosarcoma-Specific Cell Membrane on Polymeric Nanoformulation for Effective and Precise Targeting. So to begin with, why osteosarcoma or why bone cancer? Because it basically targets the age group of adolescent people, basically where that's when the bone is growing. And we need to precisely target the bone because of its nano architecture. And it's very difficult to deliver drugs or any kind of nanotherapeutics towards the bone. So my core research area involves synthesis of various biomimetic nanoparticles and studying its effect on in vitro and in vivo models. Research area comprises of, to begin with, how the lacuna of existing nanoparticle system led to the evolution of various biomimetic nanoformulation, and to study the homotypic validation, that how the homotypic or cell-cell adhesion plays a major role in cell uptake of biomimetic nanoparticles. When I say biomimetic, I mean the nanoparticles which are not immunogenic in nature, and the in vivo system doesn't elicit any kind of immune response towards it. So from the proteomics and confocal microscopy data, you can see that there is not much uptake towards the macrophages and much uptake in the tumor. Drug and nucleic acid-based targeting has also been done. And to know more about this topic, that how we employ various nanotherapeutics towards bone, you can visit my poster number 42. Thank you.
Good afternoon, everyone. I am Pratul Jain. I work under Dr. Dandapani at INSTEM. There, we employ functional genomics approach for the identification of novel gene variants with respect to hypertrophic cardiomyopathy. And with this question, we have almost sequenced now more than 300 exomes from South Indian cohort. And one such gene which we have gotten from those samples is TTL, which is tubulin tyrosine ligase gene, which adds a tyrosine residue to a detyrosinated microtubules. Now, why this is very important? The detyrosinated and tyrosinated form of microtubules, the balance between these two microtubules helps in the optimum contraction of the sarcomere. Any disbalance to this can lead to the impaired contraction. And we found with the biochemical assays what we have done, we have found that this variant is a loss of function variant. And to model the disease, we have used patient-specific iPSC-derived cardiomyocytes, where we have found that in this mutants, there is an increased accumulation of detyrosinated microtubules, which leads to mechanical stress to the cells, leading to generation of ROS, which can activate NRF2 for a prolonged time, can activate the RAS MAP kinase signaling, and also impairs the calcium handling, culminating into HCM. So for the further details, I'll be very happy to take questions, and I'll be very happy to answer at poster number 43. Thank you.
Hello, I'm Priya. I'm from ICGB. We are working on elucidating purine regulatory mechanism of adenophosphoryl transferase in E. coli. This the Salvage pathway of nucleotide synthesis, it uses the free nucleobases and converts them to the respective nucleosides. And it is an energy efficient way to synthesize the nucleosides, and hence it helps the bacteria in survive in the dormancy and develops the multi-drug resistance. This enzyme converts adenine into the AMP and maintains the level of AMP. AMP is a nucleoside, and nucleoside analogs has been extensively used in antiviral, anti-cancer, fungal, anti-parasitic, and now they are being explored in antimicrobial agent. So before going into the implication, it is important to understand the substrate affinity, the substrate preference, the binding pocket, and interacting residues of this protein. For that, we have crystallized the natural basis, the natural binding ligands with this protein, adenine and AMP. We solve the structure. During this process, we solve the unique mechanisms followed by this enzyme and the class of the bacteria. And we also studied the thermodynamic parameters of this enzyme in binding kinetics. And along with that, we also seen that the conservation at C-terminal is not only conserved in proteobacteria, it is also present in different other organisms. So how we can imply this information in designing the nucleoside analogs would be discussed at my poster. So I welcome you all to the poster. Thank you.
Good afternoon. I am Rais from Dr. Minhaj Lab at Instem Bangalore, and I'm currently working on understanding the role of glutamylation modification in regulating cellular dynamics and functions. So, tubulin post-translation modifications are the major regulators of microtubule functions and properties. And one of the modification that we are interested is glutamylation, that is addition of glutamates on C-terminal tail of tubulins. Over the years, this modification has been studied extensively using a few well-known conventional antibodies like GT335 and poly-E. But these antibodies face limitations in terms of their specificity, and also they lack the ability to be used in live cell. So what we are looking here for the tools which can overcome these limitations, and to address this, we use this general yeast display library based approach where we employ peptides mimicking a particular tubulin post translation modification, and via a series of max and fax based steps, and then turf microscopy based assays, we generated a series of set of biosensors which not only label the structures in cells which have the polyglutamylation. We could also additionally identify some unique microtubule kind perinuclear structures which were not labeled by these conventional antibodies before. So with this, I would like to stop, and I'll be happy to take any questions with my poster number 45. Thank you.
Can we now invite poster number 46 to 50, please?
Good afternoon, everyone. Myself, Ramya from Rajiv Gandhi Center for Biotechnology. So today I will be talking about nanopore sensing, where we have a nanopore placed in an artificial lipid bilayer. So when this system is placed in a buffered condition, it will allow the flow of ions across the nanopore, leading to an ionic current. Now, to the system, if we introduce a molecule that can hinder the ionic flow, and it will lead to ionic current blockages. So by studying the properties of these ionic current blockages, we will get to know the characteristic of the molecule that is passing through. And this technique has been widely employed for nucleotide sequencing, where now a USB device is available in which it contains an array of nanopores where you can add the DNA sample, and by simply connecting to the computer, we will get the whole DNA sequence. So this technique being so much much more acceptable, like this technique has been now used for other biomolecules like peptides, organic molecules, etc. So what is lacking here is an ideal nanopore, where an ideal nanopore should allow the sensing of a wide range of molecules, as well as like it should allow the single molecule detection with high resolution. So in this aspect, my PhD work focused on identifying a better nanopore tool, mainly by focusing on two biological nanopores. One is Nocardia furcinica pore in AB, a heteroligomeric pore, where we studied the controlled interaction of cyclic oligosaccharides by modulating the applied potential. And the second one is a substrate-specific pore called SIM-A, cyclodextrin metabolism A, where we studied the interaction of complex pegylated peptides, and we were able to identify the distinct conformational states of pegylated peptides. So to know further in detail, you can visit my poster number 46. Thank you.
So hi, this is Safi. Good afternoon, everyone. So I'm PhD student at Cdfd, and my lab is specialized in advanced glycation end products, which are the marker for the diabetes. So in diabetic patients, the presence of age, which we are the advanced glycation end products, get increased. So I started my work on diabetes. Diabetes is a condition which cause pain because of the so many of comorbidities. And as you can see in the first box, cardiovascular is the most prevalent in the diabetic patients. But here we have so much of treatment available. The issue is with the neuropathy, which got my attention because it is very painful condition, diabetic neuropathy. So the issue with the diabetic neuropathy was that there is no proper mechanism known how it occurs in a diabetic patients. And the second issue here in neuropathy is that we do not have any proper drug to treat this condition. So I started my PhD work to address these two issues. So in literature, I found that dyslipidemia, which is the dysregulation of lipid formation, is the main issue in all the conditions of diabetes. And in same time, there was one paper which showed that aldose reductase, which was known to modulate the glucose level in a human body, found to be involved in formation of lipid droplets in cardiomyocytes. So which attracted my attention. So then I analyzed the patient samples of DPN, which is a diabetic peripheral neuropathy patient samples. I retrieved the data and I analyzed. I found that in all the samples, I could find that AKRB1, which is aldose reductase, was modulated. So I started my PhD, which I got a base from that patient samples. And I had experience in medicinal chemistry, so that gave me a base to work on that thesis. So that is the graphical abstract of my PhD. So I could found that when I'm overexpressing AKRB1, I could found the formation of lipid droplets. So with presence of mengifrin, I could revert that phenotype. So this is how this is the graphical representation of my data. And if you want to know the mechanism, you can visit poster 50. Thank you so much.
Now, can we invite poster number 51 to 55? Please come forward.
Myself, Sadanam Manuse. I am from NCCS, doing PhD under the supervision of Dr. Srikandra Pure. Cancer is a black bile without boiling, and research is always behind the cancer set and cells not able to eradicate due to the lacking of the early diagnosis and potential therapeutic target. But the revolution in the multiomics as well as biomedical engineering nanobiotechnology can be beat the cancer without cost of the patient of the life. So here we exploring the molecular mechanism of the MZB1 in multiple myeloma malignancy. MZB1 is marginal zone B1 and B1 specific cell. So previously in our lab, found the MZB1 is highly upregulated in multiple myeloma patients found by the quantitative proteomics approach. The consistent result is found in the cell line also. So further exploring their role, can we take other therapeutic targets? So we did the knockout of MZB1 in multiple myeloma cell line. And further in vitro assay suggested that MZB1 involved in the progression of the multiple myeloma, as well as in vivo assay suggesting that MZB1 play a role in tumorigenesis as well as lung metastasis. Further, we did the quantitative proteomics of the Scamball versus MZB1 knockout cell and found MZB1 regulating the oncogene and validated by the Western blot. Further, we also found the MZB1 regulating the secretion of MMP9 and IPMS data and Western blot validation suggesting that MZB1 WTAP on MMTAC2 NCAP1 the interactor of the MZB1. Further research is ongoing. Please be curious and come to visit my poster number 48. Thank you.
Hello everyone, myself Sapna Pal from National Institute of Immunology, working under Devasi's Mohanty's lab. So my work is related to structural analysis of cancer mutations in protein-protein interaction networks and kinases. So one part of my work involves building the pipeline. So here I took the somatic mutation data from all the cancers and written a script which can map these somatic mutations onto the protein-protein interaction interfaces and the kinase proteins. so that we can find the potentially oncogenic PPIs and the mutations, oncogenic mutation present in the kinases. In next part of my work, through the molecular dynamic simulations, I took the driver mutations present in EGFR kinase and analyzed their effect, the effect of mutations on the structure of EGFR kinase. And I have also tried to understand how the fourth generation kinase inhibitor, that are allosteric inhibitor, how binding of these inhibitors to the EGFR kinase stops this effect of mutations on the EGFR kinase. So to find the better allosteric inhibitors for this EGFR kinases, we usually use virtual screening. So for that, I have developed a machine learning based scoring function which can predict the protein ligand binding affinity so that we can find the better binders. And this machine learning scoring function can be added in the virtual screening pipeline. So instead of using the scoring function that we have like Autodoc Vina and Schrodinger, etc., we can introduce this machine learning scoring function and can predict the better binders against the targets. So this is about my work. To know more about my work, please visit the poster number 51. Thank you very much.
Good afternoon, everyone. I am Seema Bhatia from NIBMG. I'm working under the supervision of Dr. Sridhar Chinnaswamy. So I am working on expression and characterization of human interferon lambda 4. So type 3 interferon are also known as guardian of epithelial. It has 4 members, interferon lambda 1 to 4. This SNP, that is RS815, plays an important role in generation of interferon lambda 4 because when delta G presents, an open reading frame is generated and it produces the P179 protein, which is interferon lambda 4. While when TT presents, it abolishes the open reading frame and a short peptide of 123 amino acid protein is present. It is found that IFNL lambda 4 and its variant are associated with multiple viral, not only viral diseases, but inflammatory and autoimmune diseases. But there are only few studies that shows the expression of interferon lambda 4, which is restricted to liver and prostate cancer cell line. So my first objective is to characterize the expression of interferon lambda 4 in human PBMCs. For that, I collected the bloods from the people and I genotyped them for IFNL lambda 4. Along with that, remaining blood, I isolated the PBMCs which were stimulated with the different stimulant and protein and RNA were performed. So from the result, I can say that while doing the PBMCs, we did not see any differentiation on basis of TT and delta G. So we go back in our overexpression system where we clone the entire gene having intron and exon. And we found that TT itself producing the protein, it's not truncated the protein we are thinking. And we found that the 3 novel isoform on the basis of that. And we named that P179 plus TT, P123 plus delta G and P179 delta G. And we found that it is on the formation of these novel isoforms are on the basis of intron. And so another objective that I work on the expression and purification of interferon lambda 4 from E. coli system, where I worked on the wild type interferon lambda 4 along with K154E, which is a high activity variant. And when we checked the activity, we found that in K154E show the higher activity. And we also tested these purified protein into the Leishmania donovani. And we found that they inhibited the Leishmania donovani in our cell culture model by increasing the ROS production. If you know more about type 3 interferon, please visit the poster number 52. Thank you.
Okay, good afternoon, everyone. My name is Shabnam, and I am a PhD student from National Institute of Immunology. So basically, I work on Streptococcus pneumoniae as model pathogen, and because of the emerging antibiotic resistance and the serotype dependency of the available vaccine, the current therapeutics are rendering ineffective against this pathogen. So we need a better and alternative therapeutic targets. To do so, I have looked at the genome organization of the bacteria and found out that it is auxotrophic for certain amino acid and relies on the respective transporter for those amino acid. So we started looking for those the transporter of those amino acid, and I have found one which I had characterized during my PhD thesis. So using different by integrating the biochemical assays and in vivo, in vitro assays, I have characterized the function of the protein as well as whether it has any vaccine, because at the end we want a better vaccine target, because the current vaccines, they are serotype dependent, and protein vaccines will solve this problem. So we have also found out whether it has any potential vaccine efficacy or not, and we have found some promising results. So we are trying to take them further. I have also found out whether it is important for the bacteria in terms of virulence or the fitness of the bacteria. So to know more, please visit my poster on the second floor, poster number 53. Thank you.
Good afternoon, everyone. I'm Shabna Ansari from THSTI. My work is related to development of therapeutic strategy against hepatitis E virus and SARS-CoV-2. So there is no specific antiviral therapy against hepatitis E virus. So in order to develop therapeutic strategy, we used network analysis to get antiviral targets, and we got 3 targets, among which we chose AKT and casein kinase 1. Inhibiting these targets in HEV expressing cells, we found there was a reduction in HEV replication. Then there was a FDA-approved drug atrovirine, which is reported to inhibit casein kinase 1, is also reported to have is reported to inhibit casein kinase 1, and in our study, it showed that it has therapeutic potential against HEV. In my next study, we try to develop recombinant vaccine against HEV. So we used 112-608 amino acid region of HEV ORF2. And using this region, we did immunogenicity assessment in biopsy mice, and we found that this VLP vaccine has more immune response in comparison to commercially available Hicolin vaccine, which is available only in China. So this could be a promising candidate against vaccine candidate against HEV. In my next study, during my PhD, COVID happened. So we also try to develop therapeutic strategy against SARS-CoV-2. Human endogenous retroviruses are retroviral elements which get integrated in human genome and get evolved with the time. So we did profile analysis of HERVs, different HERVs in SARS-CoV-2 infected cells, and we found that it was only HERV-R which was getting downregulated. And upon overexpression of HERV-R, SARS-CoV-2 infection was getting downregulated. We also found a crosstalk between P38 and ERK, and we found that this crosstalk is helping in antiviral activity of HERV-R against SARS-CoV-2. This study is published in AMBO reports last year. To know more about my study, you can visit poster number 54. Thank you.
Can we now invite poster from 56 to 60? Please come forward.
So good afternoon. I am Shikha from Rajiv Gandhi Center for Biotechnology. The topic of my research is regulation of clathrin-mediated endocytosis via BMP2K or AK1-mediated AP2 phosphorylation. So this clathrin-mediated endocytosis is a key internalization mechanisms in the cells of higher vertebrate, and it is a highly physiologically relevant mechanism. So why do we study clathrin-mediated endocytosis? Because it is highly dynamic, and over 50 different proteins are coordinating within a minute. And the AP2 is the major interaction hub, and the particular phosphorylation of AP2 found to be the major checkpoint in this mechanism. So in our study, we are trying to, but the molecular details of this process is unknown. So in our study, we are trying to demonstrate the role of BMP2K or AK1 in the AP2 phosphorylation and in turn the clathrin-mediated endocytosis. So these are the methodology we followed. And the major interesting findings of our study is that in absence of BMP2K or AK1, and we found that this both AP2 phosphorylation as well as the cargo uptake is found to be downregulated with a accumulation of clathrin plaques on the cell membrane, and the zebrafish embryos found to be defective in convergent extension defect. And the major outcome of our study is that we found that in BMP2K or AK1 mediated AP2 phosphorylation facilitate the CCP maturation. So our study have broader therapeutic interventions, like recent study identified the role of this kinase in entry of SARS-CoV-2 and HIV. So the pharmacological inhibitor of this kinase can be a potential viral entry inhibitor that prevent the entry and dissemination of these viruses. So that's all about my study. To know more about my study, please do visit poster number 56. Thank you.
Good afternoon, everyone. Myself, Shilpi Jain. I am from ICGV New Delhi. So my doctoral work was on the disease malaria, and particularly on the human malaria parasite Plasmodium falciparum. So in general, in malaria, we have a very challenging situation. We have resistance against the main frontline drugs, and with no new drug in the pipeline. Our research team is particularly focused on understanding the organelle dynamics and homeostasis. I was interested in understanding how the parasite adapted to various different niches and how it deals with the stress inside the human host. For that, I have focused on one of the stress regulator, which is prohibitin 2. I initiated my work with generating various transgenic parasite in order to understand its localization, its knockdown, and by utilizing, so basically I have studied the parasite responses with and without with this prohibitin 2 in normal as well as in the cellular stress condition. So I was able to find that during the cellular stress condition, without prohibitin 2, there is a huge abnormality in the development of the mitochondria, which eventually cause the parasitic cell death, and all that happens because of the increased accumulation of the ROS species in the cell. Also, I was able to see that in the absence of this prohibitin 2, uh, the sensitivity of the parasite was much high with the existing antimalarials. So to discuss all these findings with the data in details and to discuss the implication of this small piece of work, I would like to invite you on the poster number 57. Thank you.
Good afternoon, everyone. My name is Shruti. I'm doing PhD with Dr. Jyoti Lakshmi at NIPGR. So our lab works on ways to improve plant defense against insect herbivory. When insects start chewing on plants, there's a rapid burst of calcium elevation within the cytosol, followed by a high accumulation of J-isoleucine, which is a plant defense hormone. Also, when J-isoleucine is externally added to the cells, in the cytosol, there's a rapid burst of calcium elevation. But what causes this calcium elevation was not known, if there are channels and transporters. So we did a large-scale forward genetic screen using liminometry-based calcium imaging, and we looked for mutants that had reduced calcium elevation upon JALA treatment. And I identified a mutant called JACE1, which is a JALA-induced altered calcium elevation. I did mapping, and I was able to map the a causal gene responsible for JACE phenotype, which is a ABC transporter. Now, we also found out that this ABC transporter is able to conduct calcium. So we hypothesized that this JACE1 is able to transport JA-isoleucine from the cells to the apoplast. So without any radiolabeled JALA, we didn't have a radiolabeled JALA. So we devised a LC-MS-MS-based methodology to test the transport assay of JALA from the cells. So we found out that the knockout mutant lines of JACE1 was able to retain more JALA in the cells, which means that JACE1 is the component that is responsible for export of JALA from the cells. And so we have identified this new component. And to discuss more about the LCMS-based transport assay, please come to my poster, and I'd like to discuss this. Thank you.
Good afternoon, everyone. My name is Shubham Kumar Shah, and I'm working under the supervision of Dr. Cdfd at Institute of Life Sciences, Bhubaneswar. So the title of my talk is Aberrant CD4 positive T cells in T2DM mediate meta-inflammation. So I've divided my talk into 3 parts. If you look at the problem statement, you have meta-inflammation coming through various pro-inflammatory cytokines, and you have activation of different immune cell subsets also. So we wanted to bridge this gap between pro-inflammatory markers and immune cell subsets and how these are causing inflammation in T2DM patients. For this, when we did our research, we found out that in our preliminary data, the unstimulated cells from DM-CD4 T cells showed activation of TNF-alpha and GM-CSF. So remember that unstimulated CD4 T cells don't produce any cytokines. But in case of DM, these CD4 T cells were producing pro-inflammatory cytokines, GM-CSF and TNF-alpha, which was not there at all in healthy controls. And once we activated these cells, we found an inflammatory phenotype that was completely out of range. And TNF-alpha, GM-CSF recruited two different other pro-inflammatory cytokines also, interferon gamma and IL-17. And deciphering the signaling pathway, we found out that TNF-alpha signals through TNF-R1, activating STAT1 and 3, thus eliciting the pro-inflammatory response. So basically, we found out that in physiologically, whenever you have increase in pro-inflammatory cytokines, anti-inflammatory cytokines also come up to counter the inflammatory byproduct. But when we checked for the anti-inflammatory cytokine levels, we found out the levels of IL-4 and 10 were completely diminished in the DM patients. So IL-4 is important for fibronectin and collagen synthesis, which is also important for wound healing and to resolve infections. And this can also be one of the reasons why diabetic patients with low IL-4 and 10 suffer from poor wound healing properties and non-resolving infections. So I can discuss more about my work at poster number 59. Thank you.
Good afternoon, everyone. Myself Shubhashish Dash from NIPGR. Today I'm going to talk about the identification and characterization of anticandidal activities from plant. Next. So my research focus on the curation of the candidiasis, a human fungal pathogen. For this, I have chosen the 5 medicinal important plant. And after the screening, I found the 2 plant which have the anti-candidal activities. After various chromatography technique, I found a fraction which have the potential anti-candidal activities. And after that, we want to know what is the mechanism of this plant fraction inhibiting the fungal growth. To do that, I created the RNSEQ library of fraction-treated candida cell. After analyzing the in silico data, I found that this inhibition are mostly happening due to the hampering the cell cycle genes. Out of this, I also evaluate the de novo transcriptome with my data and found then some novel transcript. And out of this, like 900 of novel transcript, I found about 300 of transcript which are mostly expressed in the treated candida cell. And with the help of the gene prediction tool, I also found the 12 novel predicted gene which are mainly expressed in the treated candida cell. To know further my research, please come to my poster number 60. Thank you.
Thanks. Before we go to the next talk, I just want to quickly pause for a moment because Secretary Deputy Dr. Rajesh Gokhale has to get going in about 10 minutes. He came here for the students, and that's something that I want to emphasize because he really cares about student training. And many of us who have known him for a long time, how much he's passionate about science and training the next generation people. So he had a lot of other meetings, but he rescheduled all of them, and then he made time to come here. So it's indeed a privilege for us to have him here. So before we close the session, for this session, before the next talk happens, in the sense I'm not closing the session, sorry. We'd like to have him say a few words. And since some of the directors will also be leaving, we also want to present a memento for all the directors to take home, because this is indeed a very important day, the way I see it for IBRIC. And we would want to make sure that everybody goes home with this. We don't want to miss anyone. The directors who are not here, they'll be sent all of these mementos so that it will be there in the institutions. So maybe we can first give the mementos to all the directors, and then we can ask the secretary to, address quickly last 5 minutes, and then we'll continue with Dipanvita's talk, and then all the other student talks. All the directors who are there, please stay back because we are here for students. Let's not disappear after these memento that you get. Thank you. Yeah. So maybe we quickly give the mementos if you don't mind. Yeah. We might start with Taru because. Huh? No, it's okay. Come. Yeah. Come.
Yeah, that's what I'm saying. Let me give it to Ola.
Why did it? So maybe you can. We'll start with Sharmila. Devashish. Devashish. Mohanty, DD and DM. Shubhra and then DD.
Finish it.
Arvind, please.
What you do, don't open everything.
Yeah, exactly.
One
That's
only.
please. We got the idea. We got the idea. It's the same thing.
Pulok.
Manisha, come.
I'm going in the way they're sitting. You're holding it. Yeah.
Yes, sir. You give it to us. Instead of memento, give me a cup of tea.
That will happen.
If you receive memento, you get a thing.
Manisha, please. I think we are done, right? Yeah. So I request Dr. Gokhale to say a few words and then. Yeah.
I've been hogging all the time actually, and I was telling Ullas that. But anyway, I think it's a great momentous occasion. Thank you, Ullas. Thank you, Taru, for making it happen. I think DBT has some really wonderful people. You can see all the directors have been contributing to it. But I think most of all, the people who have presented today actually have been such bright and such amazing talks that you have flash talks that you give actually. I think I wish I was like this at that age. And you know, you're so clear, you're so brilliant, you're so clarity of thought process and all. I think that's what the real strength in this country is actually, and we keep exporting them very often. And you know, we want to consolidate, we want to make it bigger and better here. Believe me, the world is going to change, and this is the land where everybody is looking after for any problems, to identify the solutions for it, to be able to find ways of making it happen. It's a tough journey. As I said, the middle income trap that I showed in the morning is a tough journey, because to come out from one box and move to another box does require some bit of somersaults. And these somersaults are interesting. It's a journey that's all of us should enjoy and endeavor in life. And my best wishes to all of you. I think the BRIC is likely to be the most amazing place, believe me. And even though many of you were not admitted in BRIC, I think now you're all part of the BRIC, and BRIC Plus, what we call actually, which is even bigger organization than it is. And I think we all want to make it for the country. And I guess all of you are with us, and therefore this is going to be the very, very exciting journey future ahead. My best wishes to all of you, and thank you very much for all of you for joining here today. Bye.
Okay, we will continue with the talk. So we have the last talk of the day, and of course, whoever has a DBT welcome DBT grant, we know her. It's Dr. Deepanita Sengupta. She's the grant manager in India Alliance.
Thank you so much. Thank you for inviting me here. Before I start, I must congratulate all the students. You have taken the decision to, start your PhD. That itself is a big decision. And now we see that you are almost towards the completion, and you have presented your work so beautifully. Presenting so much work in 2 minutes is really difficult, but you have done that so nicely. Big round of applause for all of you. So I'm Deepanita, and I'm representing DBT Welcome Trust India Alliance today. I'm working as a grants manager here. And, if you have watched Three Idiots, there are very few Funsuk Wangrus, but all Farhans and Raju Rastogis who want to continue in academia. I think I have some useful information for them. So just to give you a brief idea about the organization, the DBT Welcome Trust India Alliance is a partnership between DBT and Welcome Trust UK. So we get funds from both the agencies, and then we fund in our own way to biomedical researchers in the country. So we are limited to fund only not-for-profit organizations. That's why I said that if you want to continue in academia, then only this might be useful. Sorry, did I do something? Okay. Okay. We are located in multiple cities in India. By that, I mean that, we have funded so many fellows that they are located in all over the country. And in Cdfd also, there are few fellows funded by India Alliance. So in brief, we call it India Alliance. So please, I will be using India Alliance term frequently in the rest of the time. And, we have funded more than 200 early career researchers. And that's what I think might be useful for you to hear the story about. And this is a network of collaborations. When I say collaboration, it's mainly interdisciplinary collaboration through collaborative grants. We have heard about, multidisciplinary PhD as well today. And this is like we kind of promote collaboration. And you have also heard about going out of your comfort zone. So, we try to push people to go out of your comfort zone. So for the early career researchers that we fund, we don't allow them to continue in their PhD environment. So they have to, go out of their PhD institution and find a position somewhere else. And these are the only few awards that I have mentioned here that the India Alliance Fellows have succeeded to get. So these are the various funding programs. We fund through fellowships and grants. The fellowships are mainly, like I was saying, for early career research, and then there is an intermediate fellowship for the people who are just starting their laboratories as independent faculties, and then there is a senior fellowship. Grants are of multiple categories. I will talk about that later on. But we fund basic biomedical research, clinical research, and public health research as well. If you go to the website, India Alliance website, just indiaalliance.org, you can type, and on the top you will find funding. Under that tab, there are multiple information about all the fellowship types, if you want to learn more about it. So for the early career fellowship, eligibility is the last year of PhD or till you are up to 4 years of postdoc. So that is the eligibility criteria. You can apply anytime between this. And the duration of the fellowship is 5 years. So once the grant is activated, it runs for 5 years, and it includes a total cap of 1.7 crore, and that has a salary component. Most of our early career researchers don't have a regular job, so they get the salary through the fellowship. But if someone has a job and the institute is paying them, then also it's fine. We can top up the salary, or they can use the entire budget for research. So beyond salary, there is also money for consumables and purchase of small equipment. So you can't purchase large equipment beyond 15 or 20 lakhs, but small equipment that is used regularly in the lab, that is allowed to be purchased under early career fellowship. There is one support staff allowed. One technician can be hired through the fellowship. Also, like the regular path that we see, people go abroad for postdoc fellowship, but these people, we are telling them you have to choose an Indian not-for-profit host institution to stay and do your research for 5 years. You are not losing out, because there is this provision of work outside host institution, and which allows you to go to any lab in the entire world. You choose the lab that is, matching your needs of the project, and you can spend up to 2 years. And around $3,000 per month is paid for the sustenance during this period. So this is a very important and very useful, advantage of this program. There is also travel allowance and contingency. So we don't ask why you want to use this, but anytime during the 5 years period, if there is a meeting you want to attend to, if you want to see a collaborator for a brief time, everything is allowed. And we normally don't ask back how you have spent this money. So this is like around 10 lakhs of funds, which is given as contingency and travel funds. This is kind of the cycle, how it goes. So first we receive a preliminary application, which is a very small application that includes the CV and the 750 words of abstract of the proposal. And only after this is reviewed and it gets through the next stage, you are invited to submit a full proposal. And that full application goes through international peer review. So the reviewer can be anywhere in the world who is expert in that area. And we get at least 3 critical reviews on each application. This is very important because our selection committee uses these reviews, and also it is sent back to the applicants. Whether you succeed at the last stage or not, that is, a later part. But you get these review comments from very, esteemed scientists, and that tells you where are the gaps, what are the good points in your proposal. After the applications get shortlisted for interview, there is a face-to-face interview. You have to attend an in-person interview, and then the decision is made. The post-award stage is for 5 years, like I said, and there are multiple requirements during this post-award stage. We see how the research objectives are met and how you are progressing. Of course, for early career fellowship, you need to choose one fellowship supervisor. who should be based at the host institution. So the institution where this work will be done is called the host institution, and fellowship supervisor has to be based there. So it's a mentored fellowship, but again, there is a lot of independence added to this fellowship. We support equity, inclusion, and diversity, and there is a lot of flexibility in how the funds can be utilized. There are opportunities for early career researchers through this fellowship, and there are also other platforms that we want these early career researchers to come at. For example, we have an annual conclave where all the fellows that are funded by India Alliance throughout the country, they all come together. That's a huge opportunity for networking. There are opportunities for women, and we don't calculate, if there is a career break due to multiple reasons because of sickness, maternity break, or some other reason, we don't calculate that. So those all will be subtracted from the total number of years of experience. Thankfully, we have seen a growth in the number of female beneficiaries that we have, awarded in the last 5 years compared to the previous 10 years. And these are actually this chart here shows the different disciplines that we have funded. It includes discovery research, which is the major part, the basic research that we say. And it also includes, research on infectious diseases, nutrition-related disorders, emerging health challenges, mental health, and many others. I also want to talk about the research management funding opportunities. This is a slightly, different career track where, research managers support the scientists. So for large grants where the PI has, too many responsibilities to complete the research objectives, the research managers come in there to support them with administrative responsibilities getting done in time. For example, while submitting an application and also during a grant, there are too many requirements by the funding agency. Like if you ask me from a funding agency perspective, I would like to see how the research is progressing, how much fund is being utilized, everything. And this requires a lot of work to be done to complete these reportings. So the research manager comes in here, and the research manager should have experience in science. So all the research managers working in this country are PhDs. So, they have experience of how science works. And so they can act as a good bridge between the researcher and the administration of the institution. And we support research managers through multiple grants. And there is also opportunity in funding agencies like ours. So we have a team of 9 people at India Alliance right now, but we may grow if requirements are there. So, in our team also, all the people come with PhDs and they work as grant managers, grant advisors. So I will stop here. And I have given my email and the phone number of the office. We are available over email and phone. If you call the office, someone will respond. Definitely. And if you have any, query, I'm here for some more time, so you can catch me. Thank you.
Thank you, Dikpanita. We'll now move on to the last round of the flash talks. I'm sorry, Dikpanita, we need you back here. I request now Dr. Rashna. You remember, okay. Rashna to present the moment on the Dikpanita.
Thank you, Dikpanita.
Thank you, Rashna. Thank you, Dikpanita. Now we move to the last part of the flash talk. Now I invite poster number 61 to 65. Please come forward.
Start.
Good afternoon, everyone. This is Shweta Chaudhary from ICGB New Delhi, and my PhD work is primarily centered around diabetes and tuberculosis comorbid condition. So it is reported that diabetic individuals have 3 times higher susceptibility to TB, and they report delayed resolution of infection upon treatment, and there are 6 times higher odds of death during treatment. To understand this condition, we use streptozotocin-induced diabetes in BL6 mice model, where a month post-induction, we infect these mice with MTB H37RV via aerosol, and then 21 days post-infection, we sacrifice these mice. Now, we have majorly used 3 techniques to understand this condition. We have used metabolomics and proteomics to understand the deregulated understand the deregulated metabolism in a DMTB comorbid liver, and to understand the immune landscape of a DMTB comorbid lung, we have used single cell RNA-seq technology. Now, to cut this very, very long story short, what we have reported is that in a DMTB comorbid liver, there is higher cysteine degradation and higher retinol degradation, which might lead to worsened glycemic control as well as accelerated progression of liver disease in this condition. And this we have recently published in JBC. In addition, in terms of the immune consequences of DMTB comorbid condition, we report that a DM lung shows increased tissue damaging TH17 responses and reduced anti-TB TH1 responses. This was a very bare bones description of the work. To discuss this in greater detail, please visit poster number 61. Thank you.
Good afternoon to all. My name is Shyam More. I am working under the guidance of Dr. Vasudevan Sheshadri from NCCS, Pune. So my PhD title is that molecular analysis of the translation regulation of insulin by RNA binding transactive factor. We know that whenever we take a meal, that leads to increase our blood glucose level. So that increased blood glucose level sensed by our pancreatic cells, and that in response to release the insulin. So our lab is already shown that in a response to glucose, protein disulfide isomerase PDI can bind to the insulin 5' emitter that is called as CON1 and increase the insulin translation. So this is the structure of PDI, consists of 5 domain A, B, B prime, A prime, and small C domain. So when we purify this full length PDI into the bacterial system and check the binding through the EMSA, so we observe that full length PDI cannot bind to the CON1. So it means in cell line, some post-translation modification was happened. So we try to find out this post-translation modification. So we pull down with this bacterial expressed PDI and incubate with pancreatic lysates. So we observe that in a high glucose condition, PDI can get phosphorylated. So we choose so we choose periphery probable candidate of that may be phosphorylated of PDA. So carry out in vitro phosphorylation, in vitro kinase binding assay. So we observe that 459 position can bind to the insulin 5' UTR. So in next objective, in that which kinases can phosphorylate that PDI. So for addressing that, we choose the BiID methods. So Beer ligase is fused with PDI and make a stable cell line. After that, lyse the cells and pull down the streptavidin beads and check the interacting partner through the mass specs. So further details, I would please visit to poster number 62.
So, good afternoon all. In my doctoral study, I am studying to understand the role of FBXW9 in breast cancer. So FBXW9 is a box protein, as you can see here, and it is a component of SCF complex, which is the E3 ubiquitin ligase. So it functions to ubiquitinate a protein. Whenever a protein gets ubiquitinated, it undergoes several cellular process like protein degradation, protein stabilization, also DNA repair, and so on. So our lab mainly, sorry. So our lab mainly work in ubiquitin biology and cancer biology. So one previous screening was done in our lab on breast cancer cell line, where we found that ABXW9, which is the ABOX protein, and it potentially is a potential candidate to regulate the breast cancer cell proliferation. So now the inhibition of breast cancer cell proliferation might be because of two reasons. Either ABXW9 is arresting the cell, or it is inducing the apoptosis. So in my study, I found that ABXW9 is arresting the cells at the G1 phase of the cell cycle, and we found that further study that ABXW9 is actually targeting and degrading the cyclin D. We know that cyclin D is an essential cell cycle regulatory protein which plays crucial role in G1 to S phase transition, and also it plays as oncogenic role in the breast cancer. So using different molecular biology techniques like Western blot, immunoprecipitation assay, in vitro interaction assay, ubiquitination assay, and other phenotypic assays like metastasis assay and colony formation assays and all. So we found in our study that cyclin D is getting phosphorylated by GSK3 beta kinase, and after being phosphorylated, cyclin D is being recognized by FBOXW9. Now the FBOXW9 ubiquitinates the cyclin D. The polyubiquitinated cyclin D undergoes proteasomal degradation. So this is how the FBXW9 degrades cyclin D to inhibit the breast cancer cell proliferation. So I may discuss in detail in my during the poster session. So my poster number is 63. Thank you.
Hi everyone, I'm Sonam from NIIB. For my PhD work, I am working on bovine filariasis, which is a tick-borne disease. The causative agent is Theileria parasite, and it has been already reported that after infection, it shows cancerous hallmark. Basically, 3 hallmarks, that is resistant to death, metastasis, and hyperproliferation. So in cancer, it is known that epigenetic is playing an important role. The difference between cancer and bovine filariasis is these cancerous phenotypes are these cells will go to apoptosis after treating with buparvacone drug, which is anti-thilarial drug. So we are questioning that after BPU treatment, if apoptosis is happening in transformed cells, then if there is any role of epigenetics, specifically DNA methylation for this apoptosis. So we did in our cells, we checked DNMT activity, which the enzyme which is responsible for methylation, and we found that after infection, the DNMT activity is high. After inhibiting this DNMT enzyme with the help of inhibitor, this activity is getting reduced, and mainly after after treating with buparvacone, DNMT1 and 3B is majorly affecting. So these two may be the important role during filariasis. So to know further, we did the whole genome sequencing analysis, where we came to know that the genome of Theileria infected cell after treating with BPQ is hypomethylated. Then we studied differentially methylated genes, where we got hyper and hypomethylated genes. And as hypermethylated methylation indicates inactivation and hypomethylation indicates activation, to confirm this, we did RNA sequencing and we got hyper down and hypo up genes. And when we are digging for which tumor suppressor genes are present in our cell after BPQ treatment. So one molecule we found is PML. So in during Theileria infection, this PML is hypermethylated, making it inactive, so apoptosis is not happening. And when PML is, we are treating cells with BPQ, this PML is hypomethylated and inducing apoptosis. So the role of BPQ, which is induction of apoptosis, maybe PML is the important epigenetic regulator behind that. Thank you.
Good afternoon, everybody. I am Sonam from ICGB. So I am basically working on understanding the host pathogen interaction, particularly in case of Mycobacterium tuberculosis, where it has recently been shown that when THP1 macrophages are being infected with MTB, so a huge alternative splicing pattern happens. So in that following this study, we have found by various high throughput interaction studies that there are various secretory virulent factors which can affect alternative splicing pattern in vitro conditions also. So we have found few secretory proteins which can cause particularly exon skipping when they are present in the, so make it smaller, when they are present in the in vitro system. So in that line, we are trying to understand the basic architecture and how and what is the mechanism behind this particular exon skipping event. So for that study, we are employing a very highly sought after technique, which is cryo-electron microscopy. And in that, we are purifying the spliceosomes in the presence and absence of this virulent protein. And then we are imaging them at 200 kV TEM. And we have now been able to get a low resolution structure of spliceosome. And we are now in the process of finding a high resolution structure using cryo-electron microscopy so that we have a better architecture, details, and can elucidate the mechanism how this event is happening. So for more details, please visit my poster, which is poster number 65.
Now we invite the poster from 68 and above. 68 and above.
Good afternoon, everyone. My name is Swaranga, and I'm going to talk about my work, which I have done in Dr. Arvind Ramanathan's lab at INSTEM Bangalore. So this work has been published in eLife, where we have shown that cells after chemotherapy treatment like DNA damaging drug doxorubicin become senescent, and these senescent cells actually secrete SASP and fatty acid derived component of SASP, which is this prostaglandin 15-DPGJ2. It actually covalently modifies a known oncogene HRAS, which is also a known regulator of muscle biology in surrounding myoblast, which leads to the activation and altered intracellular distribution of HRAS inside these myoblasts, which leads to preferential activation of the ERK or the MAP kinase pathway over the PI3 kinase pathway, and which ultimately leads to the inhibition of differentiation in this myoblast. And if we somehow abrogate that interaction, then we can actually rescue the differentiation. So this is my work in nutshell. And if anybody is interested, I would be delighted to talk about my work in detail over tea at my poster number 68. But before I part ways, I just wanted to highlight a couple of implications which we have drawn from my work, which are now currently in use. So as Dr. Ella said in the morning, that what are the potential applications of this work? So number one, what we are now working in the lab is that we are trying to specifically inhibit this prostaglandin biosynthesis pathway instead of pan-prostaglandin biosynthesis by specific inhibitors to ameliorate the effect of senescent cell SASP on muscle pathology. And also some part of my work has demonstrated that intracellular distribution of constitutively active HRAS also matters when it comes to regulation of muscle physiology. So now we are in the process of defining small molecule-based therapies to actually ameliorate the effects of constitutively active HRAS mutants especially in the skeletal muscle case. Thank you.
Good evening, everyone. My name is Vaibhav Kumar Nayan, and I am from THSTI, working under supervision of Dr. Amit Kumar Pandey. So basically, our lab works with Mycobacterium tuberculosis, and we are looking for pathways which will contribute to the persistence in Mycobacterium. So persistent is a condition where bacteria becomes drug tolerant, and it can tolerate even 10 times of the drugs which are used to treat the tuberculosis disease. So basically, we wanted to see whether sulfur metabolism can provide an insight into persistor formation in M. tuberculosis. So sulfur can be taken up by M. tuberculosis in three forms: sulfate, inorganic sulfur, cysteine, or methionine. So to study this role of sulfur in the drug tolerance, we prepared media which is devoid of sulfur, and we subsequently observed there was severe growth defect in the bacterial growth. total protein content was less, and intercellular sulfur was also reduced significantly. We did transcriptional profiling and validated a couple of genes using either by conventional knockout generation or by CRISPR interference. In short, key highlights were you can see that the bacterial growth was very clearly perturbed in the first panel, and there was reduced in vivo pathogenicity, and we could also reduce the MIC of the anti-TB drugs when we devoid bacteria of intersulfate. So for more details, please visit my poster number 69.
Good afternoon, everyone. I am Vandita Tiwari, working as a research scholar under Dr. Monica Gra at Nabi Mohali. So the title of my talk is Evaluating the Antidiabetic Potential of Anthocyanin-rich colored wheat on T2DM-induced rodent model. So colored wheat is rich in anthocyanins and have higher antioxidant activity. Therefore, we wanted to check the antidiabetic potential of this colored wheat. Firstly, we have checked the alpha amylase and alpha glucosidase inhibitory activity of the in vitro, and we found that the black wheat chapati, black wheat, which is anthocyanin-rich colored wheat, has higher amylase and glucosidase inhibitory activity. We have compared the results with the normal white wheat, and secondly, we have established the T2DM model through hyperdiet and streptozotocin combination. And we found that black wheat enhances the insulin signaling and glucose uptake in liver and adipose tissue, further enhancing the glucolipid metabolism. Lastly, we have also evaluated the effect of black wheat on tissue damage like NFLD, renal impairment. It helps in mitigating the renal impairment, splenomegaly, adipocyte hypertrophy in T2DM model as compared to the white wheat. Thank you.
Good evening, everyone. My name is Venkat Krishna. I'm from NIAB working with Dr. Shailesh Sharma. So the title of my PhD thesis is Identification of Key Molecular Players during Newcastle disease in various tissues of different breeds of chicken. So basically, Newcastle disease is a serious infectious disease in the poultry industry, which is causing severe economic losses. So basic idea is like different breeds of chicken, they show differences in their immune response and resistance to NDV. So the objective is to identify the genetic differences in terms of genes and non-coding RNAs, which are playing significant role in determining the host immune response and determining the resistance towards the NDV. So basically, it's a transcriptomic study. So I've taken a transcriptomic data from 4 different tissues, and using the bioinformatic pipeline, which we have standardized in our laboratory, and it is available on GitHub. So basically, we have identified the genes, long non-coding RNAs, performed differential expression analysis, and identified the functions. And finally, we have validated few immune-related genes and LNC RNAs during using qRT-PCR. To know further about my results and study, please visit my poster at number 71. Thank you.
Hi everyone, myself Vikas Fandade. I work in Nabi. So my core PhD work is mainly concentrated in plant breeding sciences. So wherein I under mentorship of Dr. Jai, we are working to improve the protein quality of our hexaploid wheat. So the common major problem with the protein is that it has a negative relationship with it. So when you try to increase the yield of wheat, so protein content goes down. So on an average, normal wheat has 9 to 12% of protein. So what we have done is we have made EMS mutant lines. So around 1200 lines we have screened, and we have identified one mutant which was higher in protein. So TAC75 was the mutant name. So we crossed it with WH1105, which was current high yielding variety. So from that, we whatever population developed, we screened it in multi location for 2 consecutive years. Out of the 350 lines that we have generated, we have sequenced 200 lines, and we developed a genetic linkage map, and we did a QTL mapping. So from the QTL mapping, we identify one region, 5.6 MB region on chromosome 2B. So which to narrow down the region, we have designed endogenous marker, which is highlighted in the purple line. So we were able to narrow down the region to 2.3 MB. So this region has hosted 30 genes. So we are currently working on the functional analysis of those 30 genes. But meanwhile, what we have done is we have identified 3 SNP from that 2.3 MB region, and we have converted into single PCR-based marker. So now we have validated this PCR-based marker into 3 different genetic population, and based on the result, I have shown one downward. So you can clearly look at. So based on the genotype, you can clearly differentiate the lines on their protein content. So I would like to discuss more at my poster at 4th floor. Thank you.
Good evening, everyone. I'm Yogesh Patidar, working with Dr. Abhijit Sardesai at Cdfd. So actually, our lab is interested in how potassium metabolism is regulated in Escherichia coli. So previously, a link between PTSP, PTSO, PTS, and phosphotransferase system, which is a paralogous system of glucose phosphotransferase system, is the link is identified has role in a potassium metabolism in Escherichia coli. So previously it has shown that the PTS and terminal phosphoacceptor of PTSP, PTSO, PTS and phosphorylase system negatively regulate a KH antiporter, which name is YCGO. So in this study, I have to learn more about the how dephospho-PTS and regulating the YCGO. So using the biochemical and the genetic studies, I have successfully shown that the dephospho-PTSN specifically interact with YCGO at the C-terminal region, and the core C is the binding site of PTSN. Whereas we have isolated various mutant of YCGO which are not regulated by dephospho-PTSN. We have also shown that the phospho-PTSN doesn't interact with the YCGO. And lastly, we also have shown that this particular protein YCGO efflux the potassium outside the cell. And for more detail, you can visit to my poster number 73. Thank you.
Good afternoon, everyone. Myself, Yogesh Sharma, and I am working with Professor Ashwini Parikh at National Agri-Food Biotechnology Institute, Mohali. So, area of my interest include structure-guided trait tailoring in crop improvement, where with the emerging trend of protein functional diversity and enzyme promiscuity, what we do generally is we look for protein sequence diversity in metagenomes. But I would like to bring your attention to protein sequence diversity in plants. For the case study, we used one LSI1 transporter, which is reported to uptake silicon in rice. So we use this transporter, and we check the genome diversity in rice as compared to other orthologs which were present in different crops and E. coli as well. So when we found that this transporter was actually an aquaporin, and for aquaporin, we are well aware that they are restricted to glycerol, or they are thought to be restricted to glycerol or water transport. But when we looked at the sequence diversity of LSI1, we found that one mutation of a bulky residue, which is tryptophan, we found that a single mutation from tryptophan to smaller amino acid led to the uptake of the silicon and arsenic kind of bulkier molecules. So in plant biology, these aquaporins have been evolved to transport many other molecules which are actually favorable for plant, including these molecules include boric acid, silicic acid, and also some toxic analogs like arsenous acid. So we did some molecular simulation studies further to understand the atomistic detail on the transport mechanism of silicic acid and arsenic acid in particular. And we've highlighted the residue. We find out the residue which were contributing mostly to these transport. And these residues were further validated by East assay for their contribution in
uptake of silicon and arsenic. So we will be editing these plants for these sites further in plants using guided prime editing, and I'll be open for your discussion at poster number 74. Thank you.
I guess we are supposed to take a break and then go for a poster session. But I think probably looking at the enthusiasm for the poster, let's not really disturb the poster session. What we do is that we'll finish the last session in a couple of moments, and then probably we'll directly go to the poster session. Then we can really enjoy the poster for a very lengthy of time. Before we conclude, we invite all the participants to engage in an open discussion on how we can further enhance and evolve this conclave in the coming years. We will commence with Ullas, Director of Cdfd, followed by I request contributions from other directors, dignitaries, and also the students.
Yeah, thanks, Yatish. Of course, again, it's been wonderful. Thanks for everyone for coming all the way from wherever you have come from, because I think it's a long way out, particularly for people coming from IBSD and even NABI. So I would like to, wholeheartedly thank all of you for your enthusiasm and putting together the work itself. All of us, we have been discussing that the best part of this is not, that we had the dignitaries and all of that, but the best part that we all feel is the scientific talks that you gave. We really enjoyed that, and at the I would like to give a big hand to each one of you for putting this together. Thank you very much. I guess things did go well, better than what I expected. I'm sure there were some gaps or mishaps, if there were any. I would like to take the blame on behalf of all the team. My team has worked extremely well. Of course, there'll be a formal vote of thanks. But just to give a perspective that I think many of them have worked nonstop over the last 18-20 hours, because I was driving them to make sure that things happen, and they were extremely driven, and they wound up all of these things. So I would like to also give a big hand to all of them. Like I said, there'll be a formal vote of thanks, but every one of them, the faculties, the admin staff, the technical staff, everyone. Again, thanks to the directors. I don't think this happens in any other organization. I've been part of many scientific institutions. At least right now, the directors that we have, I think there's some kind of a unison in terms of what we all want to do in terms of science, and how we want to make each of our institutions and the BRIC, make a mark in the Indian scientific space. It's been wonderful working with all of them, and it's again a reiteration that they're committed to the BRIC and all of you by traveling. All of them have extremely busy schedules. I'm sure you will understand that. They made time and they sat through all the talks. Again, I would like to give them a big round of applause for coming. Thank you very much. So now, of course, I'll ask if there are any people who want to say. It's not necessarily the directors, anybody whom you want to say a few words. Also, maybe please help us in giving your frank feedback on how else we can improve this, because this is the first such event. I'm sure it could have been much better. And this is something that me and Taro will take it forward, but also other directors as well. We will evolve this into an event where it should become a go-to event in the calendars of all the students and also the companies and other people who want to come. So please give your frank feedback if there are any. If there are any lapses, I would like to again take the blame on behalf of everyone that was worked. Yeah, it's open now for everyone.
Thank you, Ullas. I'd like to take this opportunity to echo what Dr. Gokhale said, that fabulous presentation, very good clarity. But one message I would like to pass to the students. Some of you would have realized when you made the presentation, your fonts have gone haywire, that they have changed. And to address that, try to utilize Arial fonts, Times New Roman, Calibri, certain fonts which are highly transportable from across one instrument to another. This is a message I wanted to pass. And secondly, if let's say you are working on Mac and in some other conference, you are going to give it on Windows or Windows to Mac, then once your slides are completely frozen, converted to either a PDF or to an image, so that it doesn't change its size and shape when it transfers from one machine to another, because that will actually upset your rhythm in a 2-minute talk or a 3-minute talk. You do not want your slide to go haywire, and then so this is just a message I wanted to pass. But excellent scientific content, no doubt about that. Thank you.
Anyone, please?
No, so I just want to thank both Ullas and Taru for putting up this. It's not so easy, in such a short time to come up with all these, and it's fantastic show, and everyone is here. So let's give them a big round of applause. And thank you so much to all the students. But like others said, it says there's always room for improvement. And I think that should come from the student primarily and not from the directors. So please feel free to give your feedback. So this keeps getting, improvement every year, and we do. One of the things which I felt is that there may be more sort of industry people, which you can bring them here so that there is more interaction. That's something I felt that we can improve on, but students can add more.
Yeah, so one of the things that we did discuss when just before Dr. Gokhale left is that we should also make sure that companies, and it's not just companies, everybody who wants to employ you should also make a case that they should attract you. It should become like you are the prized positions and they will compete amongst themselves to take you, not the other way around. So that's the hope, and we hope we'll evolve into that. Yeah, thank you, Arvind, again for making that point. Yeah, anyone?
I think joining Ullas, whatever he said, and wonderful, as one said a while ago when Dr. Gokhale was leaving. So we just, really 3 cheers to all the students who came forward. And it's totally pleasure listening all the flash talks. People were very focused and they were very clear that they had to talk for 2 minutes. But for the whatever suggestions came, one always built up and improving things. But yes, it's a beginning, as we said. And yeah, surely, Dr. Arvind, as you said, thank you for your kind words to both of us. And industry, yes, actually. So we're going to kind of join more dots in the coming one. We just thought that alternate years once Cdfd can do and NIB can do. And then we understood in the morning when secretary said, so why stay in one place? Going across is always better. So surely we'll devise a different model, which is a good model. And yes, students, it is for students mainly, and connecting them with the not only academia, with the industry. There were enough number of people. So though we don't have as many industry partners as much we wanted. So our teams, both the teams, I personally like to really applaud what Ullas said. Teams really worked very well, from NIAB, Sonu Gandhi, Sandeep, Yash, and Sanjay, and all student volunteers, and here the team Murli and others. So we were having, I think, n number of mails every day. It goes to groups, everybody knows. So it was a huge team effort, and it really went smoothly across the teams. So thank you very much, teams. And for sure, as I said in the morning, nothing as the number of students and all that. However, coming to the point, an important point is, so we wanted 50 industry and mails went. And people, we were ticking. So there was 7 confirmations till last evening, and we expected people to be here. Some came, some did come. So, and some of the industry people we have had right here, which we thought that would really encourage the youngers, the budding ones. What I like in the panel discussion, some very important points which were made. They are very, very good, and people are very clear. And I liked you when you were talking about the prostaglandin and focusing it. So it means, the message from here has really been percolated down very well. So that's a good point. And next time also we like to, as I was saying in the morning, we would like to it is kind of again a tripartite. So alumni and outgoing students and the industry, of course, academia is there. So that we look forward, and it's with everyone's input to that.
Yes,
And
students had some questions and inputs. In fact, yeah, please go ahead.
So I wanted to raise this point during the panel discussion as well, but I couldn't because of the time constraint. So for the whole day, although it was a fantastic session, but most of the emphasis was given to research. It may be academia or science, but when we talk about future career prospects, some of us, including myself, are interested more in scientific education. There I felt there wasn't enough emphasis for that because I have seen many students or even scientists, older scientists, complain that the scientific education is not at par as it should be. So if that could be considered, then that will actually really help because it's a placement conclave, so to
Yes.
speak.
Yeah, absolutely. I think, in fact, what we wanted to do, and we've been very conscious, is that, like I said, one of the constraints that we work with is that we had less than a month to put this together. So people's availability always become a nightmare.
No, I understand.
No, so
in addition,
it's a very good point.
yeah.
We also wanted to bring people in the publication industry, journals, all of that,
fell
right?
down. She fell down. She had a fracture. We were bringing scientific journalism and
People.
scientific writing people. So two people could not come. One fell down. Continue, please.
Yeah. So, no, we had, see, several of our alumni actually are, editors in Nature Medicine, iScience, Cell Reports. We also want to give, you the exposure of the kind of journeys that they have taken. So we have called them, we've called even several other publication houses. So this time we couldn't manage all of that, but we had this in mind. And thanks for bringing this up. We will definitely include it in the next one. Yeah. Any other student?
Yes.
Yeah.
Take the stage
Yeah.
for a second.
I think we all agree that
So-
this has been a beginning of what we have been doing. Thanks and kudos to both the organizers and all the directors. You know, there have been some suggestions from all of you, which definitely all of us will try to take care of it in future. But I thought of sharing one couplet with you, which I always enjoy. This is written by Siraz Fazal Khan. I'll read out for you. वो कभी आगाज कर सकते नहीं, वो कभी आगाज कर सकते नहीं, खौफ लगता है जिन्हें अंजाम से. This is a good beginning. I think we should feel happy about. We have taken a step beyond, I think, our regular career. As Ullah said, not many organizations have come forward and done this kind of thing. So I think we should all feel happy that the good beginning has happened, and we'll continue as then we, get feedback from all of you. We also realize next year perhaps we'll have more component attached to it. I think with a good feeling. Thank you very much, and I have earned my coffee now.
Yeah, of course. Yeah.
Dr. Ashwini, what you just said, and the younger one who said made the point. So actually, our ultimate aim is to how people do it. One thing is that, as Zula said, it happens in brick for sure. It's a beginning. We really want to bring in the industry here, and there's a placement cell which will be functioning from NIAB. So we would like to bring industry wherever, whichever institute we go, and that industry, you should be value to them. This is the ultimate objective, and with that we are moving on.
So good evening, everyone. So I'm odd man out, and I think thank you, Ulas, for bringing me here. So I work for pharma industry. I'm a chemist, but I could see a lot of application of biologists in pharmaceutical industry because we are foraying in different areas like antibody drug conjugates, oligonucleotides, vaccines, biosimilars, and in synthetic biology, and we are into drug discovery programs. So there is a great application of biologist in Dr. Reddy's. I represent Dr. Reddy's here. So feel free to connect some of the stakeholders like me. I can position depending on your competencies and fit for the job. And I enjoyed the event. Being a chemist, I thoroughly enjoyed biology sessions that you guys have presented. And by training, I'm a medicinal chemist, so I have great understanding and respect for your biology understanding, how things work, how your genetic machinery works and all. So there is another event that I am organizing. I'm just taking liberty of this stage, and it is happening first time in industry setup. It's called Chemical Research Society of India, um, partnering with Dr. Reddy's along with the American Chemical Society. It's going to be a big event, and the audiences are from various disciplines in chemistry, biology, and pharmaceutical sciences. Feel free to visit the website crsidrl.com, and if you want to attend, you are most welcome. Thank you so much.
Thank you.
You can flash a mail to us and we will down the line send it to our colleagues. Thank you.
Any other students? If you want to send anonymous feedback, you can do that as well. Anyone? Yes. Yeah, he wants to say, and then.
Thank you. It was a fabulous experience being here. Most importantly, kudos to all the directors, because such kind of synergy is not even seen in one institute amongst different departments. And having all these institutes together so synergetically, it was very nice. As far as the research part of it is concerned, I think we all got a snippet of what is happening in each lab and each institute, and the final year student presenting the story of their thesis. However, if we look at the middle word there, placement or an industry exposure, I would suggest one small point. If along with this slide of research, if you can have a portion of it which talks about your employability, it may be a skill set, it may be a model system, it may be your expertise which you might have earned, which is irrespective of your research. Let's say coding for a particular biological application. Startups. So those kind of points can be, communicated with the startups and the industry, and the industry which are interested may reach out to you. Thank you.
Well suggested. Appreciated. It will be taken care.
Yeah, Shubhra.
Yeah, first of all, I would like to thank both the co-host, Taru and Ulas, for a great job, and all the staff of the Cdfd and the NIAB. In a very short notice, you made us stay very happy and made the whole program very much enjoyable. So I just have one quick question to all the students sitting here. So before you came here today morning, what was you aspired to become after your PhD? A scientific path or a, industrial journey? And then after the whole day deliberation, now what is your thinking? Can anyone tell?
Yeah, I think I'm very happy that at least she's trying to ignite some of you. Please come forward. Yes, yeah.
Tell.
We want to continue the research and try to contribute the economical aspect as well as social value.
So now let me put the question very straightforward. After the whole day, how much you are convinced that also after doing good research in good labs of DBT or now break, that you can also contribute to developing economy through manufacturing, biomanufacturing, or any kind of industrial thing which connect your research skill to the societal program? How many of you, just raise hand, is now willing to move to industry? Wow, not bad. That's good. So
I thought more than 50% would raise hands.
so that was my question to the students. Now I don't know how many of the industry colleagues are here. So what I was thinking is that those of you who are with us today understood about this whole program, it would be good that if you can go around and tell all your colleagues whom we may not know, but you know, because you have, like here, all directors, we know each other. We can make a kind of a network, but you have your own network. Can you spread this word to your network? And then next year, if more industry can come and before this program happens, can also tell us where all you are looking for bright minds. And that will be easier to place and connect our students with industry need as well as our students' interest. So that's all I thought that will be a good thing.
Well said, Shubhra. Thank you so much. I think that's an important point if industry could really percolate it down.
Yeah, thanks again. That's a valid point. So anyone else? Last chance? Okay, so Yatish, please.
I guess I'm standing between you and a cup of tea, so I'll be very quick. Just will take a couple. On behalf of CDFD family, I would like to take a few moments to express our sincere appreciation to those whose dedication made this event possible. Firstly, heartfelt thanks to Dr. Rajesh Gokhale, Secretary DBT and DG Bairak for conceptualizing the event and gracing us with his presence for the majority of the day. We extend our gratitude to all the directors, CEOs, chairpersons, esteemed academicians, company representatives, and scientists who traveled from various parts of the country to be with us here today. Special acknowledgement goes to the students from various BRIC institutes whose participation was integral to the success of this conclave. I also want to express our gratitude to Dr. Taru Sharma, Director of NIAB, and all our colleagues from NIAB for their steady support since the inception of this event. Most importantly, heartfelt thanks to our director, Dr. Sullas Koltur, for his exceptional interest and unwavering support in bringing this event to realization. I extend our appreciation to our colleagues across the department, including the head of administration, estate, civil department, engineering department, transport section, canteen team, scientific communication, research management, bioinformatics team, and all other scientific, non-scientific supporting staff. I hope I didn't forget anyone whose contribution was invaluable in making this event in a resounding success. Finally, from Rashmi, who is not here actually, Deepesh, Murali, and Kuldeep, and myself, a sincere thanks to you. Thank you to each and every one of you. Farewell. So tea will be served outside as usual. Plus, please don't forget the poster session. So we have poster session at the ground floor, first floor, second floor, and the fourth floor. Please make sure to visit all of them.